Namaste students, I am JV Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Government University, Jaipur. Today I am going to discuss about the topic immunofluorescence and the subject name is immunotechnology. The topic is related to BTech, FBT 12 trimester and BTech, MTech integrated FBT 12 trimester. Immunofluorescence. Uh, what is the immunofluorescence? Uh, what is the fluorescence? First, we need to understand what is the fluorescence. Fluorescence is the technique in which we can understand the structure or amount or quantitative and qualitative analysis of something when a particular color is provided, a fluorescence is provided, and UV light is also itself is a fluorescence. So what is the fluorescence? Fluorescence is a dye which emits green fluorescence or any color fluorescence, and when it will emit some fluorescent, then it can be detected. Okay, so in the immunofluorescence, any material is detected when it emits some light. It emits some light, it's colored light. So that colored light is known as the fluorescence. And immunofluorescence is a term which is related to immuno, immunology, means the antigen and antibody interactions occur, and the interaction between antigen and antibody is analyzed quantitatively or qualitatively by some specific fluorescent light. So this detection is known as the immunofluorescence. Uh, Immunofluorescence is a technique which allows the visualization of a specific protein antigen when a particular specific antibody will bind to it. Okay, the specific antibodies are labeled with a compound that is fluorescent in situ hybridization techniques, and it will create some specific color that will maybe apple to green color, and after it will produce any color we can detect the amount, the size, and the quantity of the product. On- and the product may be any antigen, any antibody, any protein, any enzyme. Okay, but the important thing is that antibody is involved in binding to that particular color. So fluorescence is the property of certain molecules of fluorophores to absorb light of one wavelength and emit light of longer wavelengths. So for diagrammatically we can understand this. We have know very well known about the chemical chemistry in our 12th standards. We know that electrons are in a particular orbit. So in a orbit, when a electrons are there, suppose this is the electron. I must change the color. Uh, Suppose this is the electron. Okay, and it is another electron. When a particular wavelength of light is provided to these electrons, it will get excited. It will get excited, and it is in its a particular uh, orbit. But when it will absorb any fluorescent light, it will get excited and will move to higher orbit. It will move into higher orbit, but it is not its original orbit. So what will it do? It will absorb. It will emit the absorbed light. So its it will try to come back to its original orbit. Okay. So from here it will emit some light, and when it will emit a particular light, then we can detect its presence. Uh, we can detect the quantitative and qualitative analysis of particular antigen or antibody. So this emitted light is detected in immunofluorescence or any fluorescence technology. So this is the basic of fluorescent technology, in which electron will absorb the light, it will move to its higher orbit, and after coming to a higher orbit, it will emit the light. And when it will emit this light, this emitted light will be detected through the any particular detector. And after detecting, we can analyze the presence, absence, or quantity of a particular compound, a particular protein, antigen, antibody, enzymes. So it is a major basic principle. These are the example of fluorescent eye, fluorescence, and rhodomine. So fluorescence can be a particular method in which we can visualize everything through the fluorescence microscope. So the microscope which is used to detect the immunofluorescence is fluorescence microscope. This is the basic principle. Now we come to our topic, that is immunofluorescence. In the basic thing is that in immunology there is one antigen, one antibody, and one antibody that will bind to each other. Okay, so when they will bind to each other, we will analyze that they have been is particular specificity. So in the immunofluorescence, what will happen? It antigen have a particular site that is known as the epitope, and to this epitope, a particular antibody will bind. Okay, a particular antibody will bind. So what we do in the immunofluorescence, the antibody that will bind to this antigen is fluorescent. It is radio labeled. It is labeled, radio labeled. So when it will bind to this antigen, we can detect the presence of antigen, any protein. Okay, so this is the main basic. This will not maybe any only antigen. It may be any cell. Okay, any protein, any enzyme. Okay, so not only this antigen, this antibody can bind to any cell, any antibody. Sorry, any antigen, any cell, any protein, and any enzyme. So this is the basic. In this diagram, we can understand that this is the antigen. Okay, it is a antigen, and this is the epitope of this antigen. So what will happen? This antibody will bind to this epitope of this antigen. It has been bind, and another antibody that is labeled with any fluorescent tag will bind to this antibody, and this antibody is specific for Fc portion of this antibody. So when it will bind to the antibody, first antibody, then it will uh, emit some light which can be detected, and which can detect the presence of some particular cell, some particular kind of antigen, some particular kind of proteins. So this is the basic diagram. this is the antigen antibody. Okay, it is an antibody. This antibody will bind to antigen. Okay, this is the paratope which will bind to the epitope of this antigen. Okay, it is our first antibody. So what is the basic thing is in the immunofluorescence, that any anti-antibody will bind to this antibody. Okay, but in the immunofluorescence, anti this anti-antibody is labeled. It is labeled. So this labeled anti-antibody will bind to this antibody, which can detect the presence of a particular product, antigen cell, or any protein. So there are two basic principles of immunofluorescence, and two ways are there: the direct immunofluorescence and the indirect fluorescence. In the direct fluorescence, only one antibody is involved. Okay, and this single antibody is radio labeled. Okay, the Fc portion, this is the Fc portion, and this Fc portion is radio labeled, and the single antibody will be used for the detection of a particular kind of epitope. What is the advantage of direct immunofluorescence? This is technique which have advantage that it can combine the antigen directly. And what will be the benefit of this? That it will reduce the steps, not more steps will be there. So it reduces the steps, and maybe the process may be the faster. What is the indirect immunofluorescence? In the indirect immunofluorescence, we can understand by the diagram. Similar thing is that this is the antigen, and the antibody will bind to this antigen epitope. Okay, it will bind to it. Here it has been bound. Now what will happen? Uh, the secondary antibody will come here, which is labeled to a radioisotope. Okay, this is labeled to a radioisotope. So this anti-antibody, labeled anti-antibody, will bind to Fc portion of this antibody. So it has been bound here in the third step, and when it will bind to this antibody, then it will emit some light, and when it will emit the light, then it can be detected from the fluorescent microscope. Only fluorescent microscope can detect this uh, emission. Okay, so in the simply we can understand that this is the antigen, and an antibody will bind to it, and to this antibody another and secondary anti-antibody will bind, which contain a fluorochrome. Okay, a fluorochrome will bind to this antibody, and when it will bind to this antibody, it will emit the light, and after the emission of the light, we can detect the presence of a particular kind of antigen. So what is the advantage of indirect fluorescence? Uh, the anti-immunoglobulins can bind to each antibody present in the first layer. The fluorescent is brighter than the direct test. Okay, so the light is more lighter, more darker. Okay, it is more clear than the direct. So first advantage is that the light and the picture is more clear in indirect fluorescence. It is also more time efficient. Okay, take less time. Okay, the second advantage, and a different primary antibody is used for each target protein. Variable part of primary antibody bind to specific part of the target protein. So what is the next advantage? That a particular kind of antibody will bind to a particular kind of protein, that is the specificity, that will increase the specificity, and the secondary antibody will bind to the constant part of the primary antibody, and therefore a sample of the same batch of the secondary antibody can bind to many different primary antibodies. And what is the advantage of the fourth, that it will bind to the constant region, not the variable. So this is the constant region, and the secondary antibody will bind to this constant region, not the variable region. So more and more antibodies will be can bind to this antibody. Okay, more and more anti-antibody can bind to this antibody because it is have the ability to bind to only the constant region, not the variable. Only one antibody can bind to this variable region, and constant region can be more for the binding side. These are the pictures of fluorescence, which can be which taken from the fluorescent microscopy. What is the applications of immunofluorescence? Immunofluorescence can be used in the tissue sections, culture cell lines, or individual cells, and it can be used to analyze the distribution of proteins and small biological and non-biological molecules. Immunofluorescence can be used for the combination of other non-antibody methods of fluorescence stunning. For example, it can be used for the detection of 4,6-diimmunodiol diol, and the technique has a number of different biological applications, including evaluation of cells in the suspension culture, tissue beads. It is important in the diagnosis of autoimmune disease. So in this lecture, we have understood what is the immunofluorescence, and the use of this immunofluorescence will be covered in our next lecture, that is fluorescent activated cell sorting analysis. So our lecture is over here. We will meet in another video tutorial. Thank you very much.